Hello and welcome to the Interacting with R Shiny Apps video module, which is a part of the 7 Bridges onboarding lesson. While watching this video, please keep in mind two key concepts. The CGC has several interactive web apps in the form of R Shiny applications. These interactive apps are suitable for many different types of data analysis. At the end of the video, you will be able to 1. Navigate to the interactive web apps page, and 2. Understand how to analyze data while using these apps. In order to access the interactive web apps, you'll need to follow these 3 steps. First, click on Public Apps in the main navigation. Then, choose the option Interactive Web Apps. Lastly, you'll click Open for the app that you want to access. For our demonstration today, we will do each of the following. 1 is navigate to the interactive web apps gallery, and 2 will explore some of the R Shiny apps hosted on the CGC. The first item I'd like to show you is an R Shiny app called Circos that generates high-quality circular plots for visualizing variations in omics data. This was originally developed by Ying Hu in R, and this tool was adapted for the CGC for an easy-to-use point-and-click interactive app. To get to Circos, click on Public Apps along the top blue banner, then go to Interactive Web Apps. From here, you'll be taken to a new page with some of our R Shiny apps. We have several to choose from: the dbGaP Submission Form Suite, Genesis Model Explorer app, Locus Zoom, and finally, Circos. Click on Open. From here, you'll be taken to a new splash page. Click Yes to proceed. So this is the Circos main splash page. We have a user guide available, as well as references that use this plotting tool. Once you're ready to start, click Start Discovery. So for Circos, the first thing you'll do is input data. You can choose from your own data from several of your projects, or use packaged built-in data. Today, I'll be using the built-in data. So I'm going to select this to turn it on, and then Load Data. Click on Load Data, and a pop-up will appear saying Data Loaded. Next, we'll select Mapping Data. And again, for this data, I'm going to use the built-in data. From here, I'll select maybe TCGA, BRCA, expression data. Once ready, click Load Data. I'm going to add some link data as well. So for this link data, I'm going to choose the TCGA, BRCA sample names, and subtypes. Then click Load. Once ready to create your plot, click on Visualizations. There are several plot types available, and you can stack these. To start, let's click on the chromosome plot. So it'll ask you to choose segmentation data, mapping data, and a column from mapping data to plot. So I'm going to choose the loaded data that we selected. For mapping data, I'm going to choose the gene expression, and then I will plot name. You can modify the circle radius or width, and then choose specific colors. I'm going to use the palette picker and use a rainbow palette. Once ready, click Submit, and a new plot is added. If you'd like to visualize this, click Generate Plot. From here, we can build upon this and add more information. For instance, let's click on Heat Map. Again, we will use our same data types, and then again, I will use chromosome to plot. Using the same palette picker, I'll click Submit, and once again a new plot is added. To generate this new plot, I'll click the button, and both the plots are overlaid on top of each other. We can modify this to visualize it a little bit better. So let's change the circle radius to something smaller, like maybe instead of 400, we will choose 300. Click Submit, and let's regenerate the plot. And now you can see two distinct plot patterns. From here, we can keep adding different plot types using the same format. To add link data, click on Link and load the selected data. Then go to Additional Settings along the bottom, and you can choose line width, whether you'd like to increase the font size, background, or include labels. Let's turn labels on for this example. Oops, forgot to select our color. You could add as many or as few plots as you'd like. At any given moment, you can edit these, or go to General Plot Settings to give it a name. So let's say Tutorial Plot. You can set a title for your X and Y axis and change the aspect ratio. Once ready, click Refresh Plot, and you'll see the name appears. You could add legends or labels. A little bit hard to read, so let's change the radius of this and regenerate this plot. Next, let's talk about the dbGaP submission form suite. Once you've completed your analysis, here's a tool for you. This tool streamlines a way that a CGC user can submit their data to dbGaP. Users can easily export the metadata from a CGC project into this tool, then transform it using the user interface to match the dbGaP submission form. Once the data is transformed, all that's left to do is download the Excel file and upload it to dbGaP. Once again, it's the very first option. Click Open to load a new splash page, then click Yes. Once the page loads, you'll see two options: Need Help or dbGaP Form Creator. On the Form Creator page, you'll just follow the prompts to get your data ready for dbGaP submission. So as you can see, you can select which project your data will be found in. Once you specify that, you'll be asked to load data, so you can select the file and then load. And then you'll get a notification that it's finished, and you can move on to the very next step. Once your data is loaded, you'll get some summary information along the bottom here. We have a column with gene names, as well as some generated measurements. You can interact with this, copy, move things around, and filter as well. Once you scroll down, you'll see kind of a summary of the file. Click Next along the bottom to proceed. And once again, just fill out the information and follow the prompts. The next app I'd like to show you is called Locus Zoom. So the immediate button that you see is a user guide. However, you'll see a tab along the top here with many options. One is a user guide. The home page will just be this, what you're seeing. This is a useful tool because it has built-in data. So it has example data sets from the University of Michigan. So you can explore those easily, or you can use your own data sources as well. I'm just going to show you what we have built in currently, but this really helps for future integration with more, EPI kind of data sets. And as we integrate the CGC with SAS and other things, this will be a very useful tool. But I've just clicked on one of these data sets. I can explore which chromosome I'd like, specify starting position or any kind of flanking regions, and I can mess with these and kind of adjust it as I need. But from here, you'll see a plot is generated.
And this shows log transform p-values along the y-axis, and you'll see the chromosome along the bottom. So this is a plot for linkage disequilibrium, and it basically just compares SNPs to the top SNP. The top SNP is represented by this purple diamond, and everything else here with a value for the LD is shown in blue. If it's gray, it doesn't have a value. So really, this is a useful plot because it's very easy to generate. You can save as a PNG or SVG. You can show legend or remove the legend however you want. So it's a really useful interactive tool.
Some key takeaways after watching this video. 1. The CGC hosts over 900 tools and workflows in our public apps gallery and several RShiny interactive web apps. 2. Users can dynamically change inputs, manipulate data, and instantly see the corresponding visualizations or results using these web apps. Our next video in the module list is interacting with Data Studio environments, specifically RStudio.